Thank you. So I was asked by Margareta to provide some scientific background to the problems of dual use and the transmission of highly pathogenic avian flu. And at SVA, the National Veterinary Institute, we are the coordinator of a large EU project called ANI-Biothreat that are working with the development of methods to combat bioterrorism. So this is quite a big issue also in the EU as a whole. But in this presentation I will talk about these controversial transmission studies that has been debated since early this year, the results by Fouchier and Kawauka. But I will start on a very general level. Since I'm at BMC, I'm pretty sure that most of you know very well this slide. You know that we have the general molecular biology scheme is that you have the genes. So we have this, the general scheme that we all know about is that we have the genes on the DNA level, and then you make a copy of a certain gene that should be expressed. You transcribe, and the transcribed copy then becomes a protein. But the viruses, they have come up with some other additions to this general scheme. For instance, you have the reverse transcriptase that goes from RNA to DNA. But for us, the most important thing is these RNA-dependent RNA polymerases because they are used by viruses, in particular the RNA viruses. So we have the RNA-dependent RNA polymerases. But they generally don't have proofreading, and they have a quite poor accuracy. And this means that you will have a very high variability of the genes that the viruses have. So you get some kind of quasi-species. You have a whole population of different viruses that can very easily adapt to new circumstances. So you have then you can have both the negative sense RNA and the positive sense RNA, and these are the RNA that can immediately become a protein, while this is the complementary part of that. Okay, so now we will talk about negative strand RNA viruses. You have this is a very important group of viruses because they cause many diseases, important both for animals and for man. For instance, you have the Lyssa viruses that cause rabies. You have the Morbilli viruses that cause measles. You have the Rubella viruses that cause mumps. And the important poultry pathogen is the Avola viruses that cause Höngspest, that is Newcastle disease virus. And of course the filoviruses are very important as well. You have the Ebola and Marburg hemorrhagic fever causing viruses. So these are the mononegavirales. That means that they have their genome in a single long DNA molecule, RNA molecule. But then you have this group, and that is of big interest to us. Here you have also several important viruses. The hantaviruses, for instance, cause sork fever, and the arenaviruses are also important human pathogens. But the Orthomyxoviridae, that is our particular virus here, the influenza A, and here you then have the H5N1 virus that we will discuss today. So these viruses, they have a segmented genome like this. There are 8 segments, and the most important one for us, and very important in this virus, is the hemagglutinin. It's the 4th segment. If you order them by length, it's a 4th segment. So you have the hemagglutinin gene, and the hemagglutinin protein is on the surface. It's the surface glycoprotein of the influenza virus that gives it its serological properties. because it is exposed. So you have the hemagglutinin and you have the neuraminidase. These are the two. So when you hear about H5N1, that means that you have a fifth type of hemagglutinin and you have the first type of the neuraminidase. So we have then the important properties of the hemagglutinin is that it accounts for the binding to the cell. It gives you the host specificity, and it is also important for the release of the RNA into the cell. And another important thing for us here is that it has to do with the the stability of the virus particle, as you might imagine, since it covers the whole particle. So this is a very important protein for the general properties of the avian influenza. So let's take a little bit closer look on it. So this it is actually trimere, but this is a monomer that I show here for simplicity. So the two important things for our discussion here today is first, the receptor binding takes place up in this region. So you have here is a sialyl lactose that is a receptor binding molecule of this virus. So it's in this region, and that gives the host specificity of the virus. But then you hear all about the high pathogenicity of the H5 and H7 subtypes, and that has to do with the cleavage properties. This particular crystal structure shows you hemagglutinin that has been cleaved, so you actually see the ends of the two parts that are created after cleavage here. And the thing about H5 and H7 viruses is that it can become hypercleavable, so they can be cleaved all over in all tissue of the host. So it can then spread all over into the brain and in different organs. For some reason, it is only the H5 and H7 subtypes that can do this. And actually, I have a project together with a group here in BMC where we are trying to look on this problem by informatics approaches. So the great diversity of this virus makes it able to adapt to various hosts. The native host is actually wild waterfowl, and then it has various subtypes. You can see in black here is subtypes that has adapted to the particular host. For instance, the seals, you have H7N7, H4N5, H3N2. But they all once originated from wild waterfowl. And in human, you have the H1N1, the H2N2, and the H3N2. These are human influenza variants that we have seen so far. And I think this H1N1 and the H3N2 are the two circulating now, and we recently got a new H1N1 variant, the so-called swine flu. And the vaccines that we take, they are actually combination vaccine where you take into account both these subtypes. But as I mentioned, they all can be found in the native host almost. But we have actually the H16 subtype appeared in Sweden in 2006. It was found in black-headed gulls. And very recently, just a couple of months ago, the H17 subtype appeared in yellow-shouldered bats in Guatemala. So this is a moving target. It's this virus adapts to different hosts, and it is and that is partly due to these properties of RNA viruses that I mentioned. And then we have in red here, you can see then that H5 and H7 viruses have, unfortunately it is not so easily seen, but the H5N1 viruses normally appear in chicken and in domestic poultry, but comes from the wild waterfowl, as I mentioned. H9 has appeared a lot in quail, from where it can spread to humans. It's a very zoonotic variant. H5N1 has been found in cats in Germany, for instance. And the swine is also thought about as a mixing vessel where you can go from various places to swine, and from swine it can adapt so it also can spread to humans. And the swine flu was an example of this, of course. So, but the H5N1 virus that has very serious consequences has appeared in various hosts, as you can see here. So the adaptation now that we are talking about and that this study has been working with is then because the thing with H5N1 viruses coming from birds is that it cannot easily spread between different humans. And the reason is probably related, although this is a simplification, in the fact that a man has this kind of receptors, and you have, this is sialyl lactose. Here you have sialic acid, also called uramic acid, and here is then lactose. And if you move this bond to sialyl lactose between these two oxygens, then you get two different variants of this sugar, and then one of these are typically then found in the respiratory pathway of men, while the other one is found in the intestines of birds. So this is actually found in the intestines and gut of birds, and that is a normal way that this virus are replicating and spreading. And among waterfowl it is probably spreading through the water route, because in dabbling ducks they are drinking water, so it is spreading in this way, not as in man as a respiratory disease that can be spread through air. So this is the two different ways that this virus spreads in humans and in birds. But why then is this virus so important and these results so controversial? So this is a summary of the cases in humans where you have infected men and the number of deaths. And here you can see that the total number of infected humans up to 2012, this is January to 2012, was 578, and the number of dead among these were 340. So it's a death rate of 59%. So it's an extremely dangerous disease if you become infected. As a matter of comparison, I saw in SMI's report about the swine flu, the number of cases in the age group 15 to 39 years were 520. That is not so far away from this, and not a single one died of this. So this is a much more severe disease. But the other thing that you can notice from this is, of course, that this is a time period of almost 10 years. And in 10 years you have 600 cases. So it's extremely rare that humans become infected. So you have these two things, that it is very rarely so that anyone gets infected, but if they get infected, it is very, very serious. And the importance then of the transmission studies that have been done is, of course, that they create a virus that may be spread much more easily between people than while you may have this very high probability of people dying. So then you actually get in an extremely dangerous situation, and this is why this reduction decision or recommendation came about, of course. Okay, so did I skip something there? No. Now it is difficult to know exactly how these experiments have been carried out, because they haven't been published yet. So it has just been some pieces made available here and there, and so, but this is probably relatively correct what I show you here, and these are the two guys that are heading the research groups that have published this. It's Yoshihiro Kawauka and Ron Fauchier. Actually, Ron Fauchier was also one who discovered the H16 subtype. Okay, so what Kawauka did was then that he combined the human H1N1 virus. I think this was a swine flu virus. He combined and infected with an avian H5N1 virus, and infected with two different viruses, then a ferret, Iller, and then he got a reassortant of these two viruses where all genes in the background were then human adapted genes, while the hemagglutinin came from the H5N1. So this was a reassortant virus, and then from here he went into reverse genetics work that I will describe on the next slide. So this actually will soon be published, I think, in Nature, because there has been clearance and it can be published. I should also mention that these ferrets that got then infected by this virus, that by reverse genetics could be made to spread between ferrets, actually these ferrets didn't become more sick than if they had been infected with well, why use ferrets? I should mention that ferrets actually get colds and influenza in the same way and with the same symptoms as man. So if you have a ferret as your pet, then you should be aware of that you can infect the ferret with your cold and also with influenza. So that's why ferrets have been used in these studies. So it is quite likely that if the ferret gets sick, then also humans should have the possibility to become sick from this. Okay, but these ferrets, they were lucky because these viruses were not more dangerous than the usual influenza virus. In Fauchier's study, they made a quite common way of making experiments in viral research, that you do something called can anyone serial, but yes, serial passages of the virus, where you start with a virus and you infect the first ferret, and then that virus adapts a little bit and you infect the next one, and so on. And I think that Fauchier did this 9 times, and finally he ended up with a virus that could spread between ferrets, and also, unfortunately, this virus was highly pathogenic, and the ferrets died from it. And the difference was that here he started with an H5, and completely H5N virus, where all the genes were from the H5N virus. Up here, 7 of the 8 genes came from the human virus. So that was a difference. Okay, so this then describes the details that have been public so far from Kawauka's study. He then started with an H5. He, as I mentioned, took all the genes except the H5N1 from from a human H1N1 adapted virus, combined that with the H5 gene from an avian influenza, and then that virus couldn't infect the ferret, and no one of these ferrets got infected, and it couldn't be airborne transmitted, of course, because it didn't appear here in the first place. But then by his knowing about the details of the hemagglutinin receptor binding position and the different amino acids that you can find in human and avian influenzas, he made 2 mutations, and I think they had 6 ferrets, and one of the ferrets actually got the typical cold symptoms, and then they sequenced this virus, and they found that one more amino acid change had occurred here. So it mutated, and that mutated variant could then infect the ferret and give the symptoms. But still, there were no airborne transmission. And then they introduced, again by an educated guess probably, a fourth mutation. And this virus could infect the ferret and also spread to all 6 other ferrets through air. So here you actually have the controversial data listed. So this is the experiment then that has been carried out and caused this controversy and couldn't be published. But apparently now the authorities has changed their mind, and I think both studies will soon be published. Okay, I will end by going through a little, some few remarks that has been that I have seen in the literature, and I think that most people in the scientific community would agree. That the H5N1 virus is of course a very dangerous virus, and we cannot just stop and investigate the requirements necessary to have a transmissible virus between humans, because it might happen in nature, and that I think is a general thinking that you have bioterrorists, but still you have a nature that causes this kind of problems all the time. And we have had this swine flu, and it is by no means impossible that nature will give you this kind of virus. And if you don't have any, if you are not prepared by knowing which viruses can be potentially those that evolve the property to spread between humans, then of course your preparedness will be much less effective. So that, and in fact these comments are from Kawaoka mainly, and apparently there are circulating H5N1 viruses that has a few of these mutations already. And if we didn't do this kind of research, we didn't know about it. So obviously it is important for preparedness to know about the mutation necessary and if they are out there. And also, if you want to create vaccines against these viruses, it is important to know which properties that they have. So these are simple arguments. The other one is, of course, that a practical argument is if you want to restrict this information to certain scientists and those that actually should know, that seems to be practically very difficult to do something like that. So, obviously, if you restrict the scientific knowledge like this, then the efficiency of the science will definitely decrease, and we will not be able to prepare in the best way. In particular, in the countries where you don't have a very strong internal science situation, then it is all the more important that you have access to this kind of information from the countries that can produce it. And the very same countries may also be the ones where it is most likely that these kind of outbreaks may happen. For instance, Egypt, Pakistan, then they have the avian influenza is actually endemic in these regions. And if you remember from the table, a lot of people are actually dying today in these countries. So obviously these are also the countries that will take the most harm from a virus that can spread between man. Okay, that was what I had intended to say. Thank you.